Hi everybody, this is Rebecca and our group is presenting reducing excess ammonia in cow feces and urine with urease inhibitor. Nitrogen is very inefficient in its metabolism in the cow rumen. Ammonia and urea are forms of nitrogen in the cow, and we want to increase the feed efficiency and the usage of this nitrogen by preventing the conversion of urea into ammonia. Urease converts urea to ammonia. We want to use the gene CODY to create a repressor that will prevent the transcription of the enzyme urease. Since urease creates excess ammonia in the rumen, the repressor will help decrease the amount of excess ammonia. In this way, decreased amounts of urease will slow urea hydrolysis to ammonia in the rumen. This will ultimately increase the efficiency of ammonia and urea utilization within the cow rumen and decrease waste of ammonia in the cow's feces and urine. By integrating this urease repressor in the cow rumen, the cow will increase in feed efficiency and decrease in nitrogen output. This will allow cattle farms to save money with cattle feeding while decreasing nitrogen soil runoff and improve air quality.
So since we want to target the urease, we want to implement the CODY gene as mentioned. So how the CODY gene represses is it codes for a transcriptional repressor that directly binds to the major promoter as seen in the diagram on the left. And since it binds to the major promoter sequence, the RNA polymerase cannot get to the sequence and it cannot transcribe the urease operon. So the urease will not be made, and then there would be less conversion of urea to ammonia. Next, we'll be talking about how we're going to be cloning this into the cow rumen. So in order to have the recombinant E. coli that can inhibit the urease enzyme, we're going to implement the CODY gene from the B subtilis to code for the CODY repressor. So in step 1, as seen in our cloning mechanism, there'll be digestion of the vector plasmid, and our specific vector plasmid will include the LAC operon, the GFP gene, and a multiple cloning site within the LAC-Z sequence. So the plasmid will be cut by 4 specific restriction enzymes, from this multiple cloning site, and they will cut out the LAC-Z gene and the LAC-I gene. So in the next step, step 2, there will be PCR amplification, and this will amplify the CODY gene from the B subtilis. So it's important to remember that the primers of the sequence will be engineered in a specific way to utilize the same restriction enzymes that was utilized by the vector plasmid. So that carries on to the next step, which is ligation. And so the ligase can come in and connect the plasmid and the CODY gene so we can have our final product. Step 4 will be where we can actually implement the plasmid and transform it into the competent E. coli cells. Step 5 would be plating it on a LB plate, and the colonies that are successfully transformed will be present as green and fluorescent because we utilize the GFP gene. So the GFP gene, if it is successfully transformed into the cells, will appear as green and fluorescent under UV light. And step 6 will be where we implement the cells into the cow feed, and this will allow for the E. coli cells to eventually travel to the cow rumen and take action, and hopefully we are hoping that there will be less excretion of ammonia waste with the implementation of our gene.
Okay, by adding the CODY urease inhibitor to slow down the production of ammonia and increasing ammonia utilization, we will decrease the amount of contaminants that are secreted out. Doing this does come with some risk for the health of the cow. First, by slowing down the hydrolysis of urea, we risk a buildup of urea inside the cow rumen, which can lead to ammonia toxicity. However, we hope to only inoculate the E. coli cells into the rumen in small increments. Despite the risk, it ultimately creates a lot of benefits for the environment. Adding a urease inhibitor would lead us to our goal of reducing ammonia lost in the urine and feces and improving cow urea utilization. We would reduce the damaging runoff in soil that leads to pollution of water sources and air quality. Our idea is feasible and can be implemented if we are careful about the amount of inhibitors we use and make sure to stay at a safe level. We will also add probiotics to prevent the inhibited growth of bacteria that would be affected by decreased urease activity. The Food and Drug Administration may restrict the work because there would need to be more standards put into place to include the alterations from the experiment. Overall, we believe our project would make a positive impact, and we hope we receive the funding necessary to carry it out. Thank you for watching our presentation.